Much for listening.
Thank you so much. That was so interesting, Elizabeth. I have a few questions if that's okay with you.
Yeah, definitely. Yeah.
So one of them was about the intranasal method for injecting like insulin. Yeah, I think that I guess I have questions about like how it specifically reaches the brain versus other parts of the body, because you mentioned that you noticed that blood glucose didn't decrease, but at the same time there was one other metric that you used. I forgot what it was, but you were able to tell that basically the insulin had specifically reached the brain, and like also how it's capable of even crossing that blood-brain barrier through the nose, because I'm assuming that we're smelling other things in our lives. Do they also reach the brain, or how's it specifically insulin that's going? Yeah.
Those are excellent questions, actually. So the insulin does reach the brain through this, what's called a trigeminal pathway. It's pretty much, going through the nose to be distributed through like the choroid plexus. But the reason I think that it's too big of molecules, really, what's being going up through our nose, and our nose is already filtering out these big molecules. And that's why you kind of have some what's the word, like kind of dripping, like if you get an infection, like your nose runs, and it's because it's trying to keep those particles out from going up the brain. So we're not just constantly, I mean, this is something that's being like shot up and it's going really far in. So you don't have to worry about everyday sense going through, but I will say there has been documented infections that have happened from people doing, I think it's called a neti pot, I can't remember, but it's pretty much when you have an infection, and some people will try to kind of self-heal it by putting water up there, and if that water is infected, because it's such a vulnerable area to the brain, you can basically infect the brain. So it is important to be cautious about shooting anything up the nose. But we also, the reason, and that's actually a really good question, the reason that we're not getting the effects in the periphery is because it does go to the brain, but if we were to just be giving it to the periphery of the body, like people with diabetes do, it's just getting used up in those periphery kind of task of lowering the blood glucose. Now, over time, it's likely going to sensitize that transport mechanism, but as long as there's kind of that extra adiposity and it's taking care of the periphery, you're not really getting that central. So I think over time, if that system gets normalized, it starts affecting the central insulin, but not yet, if that makes sense. Yeah, but those are great questions. Yeah.
So essentially you have to use like a certain, I'm guessing you use a lot of insulin when you're injecting it so that it can like cross through the mucus and everything else in their nasal epithelium.
Yeah, well, for this one, for the intranasal, it's actually like not, well, maybe it is quite a bit, but it's 16 puffs total, and it does take 8 minutes to administer all of it. So it is quite a lot that we're giving them. That seems to be the most common therapeutic dose. Yeah.
Cool. Yeah, that's super interesting. And then I had another question about like the insula that we talked about earlier. So it's also it's known to be like the primary gustatory cortex and like involved in like other sensory integration. So yeah, would it be like, I think it was interesting to see that somehow overactivation of the insula causes deactivation of like food choice. That would not have been my like first guess. Do you have any like explanations for that? Because if someone told me like, okay, the insula got overactivated, I would think, okay, wow, you have an increased ability to now like process and integrate like the different like taste senses and everything associated with food. So any like, is there an explanation for that, or?
Oh, I know, it's so tricky. That's actually a good point, because I think we were surprised when it was overactivation. And I mean, at the end of the day, you can't answer every question, but I suppose that's the benefit of coupling this, like the brain neuroimaging, and not just doing, so if we didn't do the task and we just kind of took images of the brain, it would be MRI, but the functional fMRI aspect allows us to kind of see it over time. So they're doing this task at the same time, so we're able to couple the changes with the brain with the behavior. So they're rating the foods as less palatable as well. So that kind of gives us an indication of speculation a bit, like, okay, this overactivation might be a good thing. And in this incident, the overactivation might be good because it's allowing them to kind of maybe hone in more to those interoceptive signals about the body and what's happening, and it's allowing kind of that increased attention to knowing that these nutrients have been consumed and you're full. So I mean, it's tricky. I think that's a good question, and you just kind of have to have a level of speculation, but it's good to have your behavior task combined with it to offer more support in that direction.
Yeah, certainly. Is it possible that maybe like the prefrontal cortex was also overactivated at the same time? Like, did you guys notice that it was specifically the insula that was most activated, or were there other parts of the brain too that
Yeah,
were?
so we did look at the prefrontal cortex because we were expecting these changes during, with cognition, and we didn't see anything during that task. So we do have another task that we gave during the scanner that's an inhibition task, and that data hasn't been analyzed yet. So it could be that the PFC was more activated during the inhibition task, but during this ratings, it wasn't yet. But that was what we kind of thought would happen, and it didn't. So it's a good point.
Okay, I'd be very interested to see like whenever you guys analyze that and that comes out. Yeah, that's super interesting. And then I had one. Do you mind sharing your summary slide again? I think I had one more.
Oh, yes, of course. Yeah.
I'm trying to recall what it was.
Yeah. Okay. And here we go. Oh, there we go. Yeah.
Oh, right. Yeah. So somehow the insulin injection seemed to cause a statistically significant difference in like desire for food prior to eating versus after eating. So is it because there are like homeostatic mechanisms that say, hey, I've eaten, I don't need to eat more? Like, is it like literally like the leptin and other like body, like peripheral signals that are coming in and crossing and signaling to the brain, like the hypothalamus, for instance, that we've eaten, that perhaps the person doesn't like that the insulin can't cause a significant change? Or what would you say the explanation for that is?
Right. So it's interesting that so in previous studies, they weren't seeing effects in women if they hadn't received food yet. And it was only seen if they had consumed a lunch before, which is why we did it this way. And I think that the reasoning behind that is because, and this is so speculatory because there's not been a study to be done, and they are showing some sex effects. So they might be interacting with estrogen as well. But I wonder if it's because there's a documented increase like food reward for women, and especially for people with obesity, they tend to obviously have more food reward. And so I think that if we think about insulin's mechanism in the body, it's pretty much to signal that we've consumed food, and that way we stop eating. And it's kind of like this satiating, it lets you know that you've eaten. So I think for people with obesity, the reason that it's even more effective if they've eaten already is because it's kind of sending that double hit of like, okay, you've eaten, and now you've also received this insulin saying even more so that you've eaten. So it might just be that they needed like more of a push to kind of bring about that knowledge that they've consumed, so food's less rewarding. And I think that's why we're seeing it only in people with obesity, but it's tough to say. And I don't know about the interactions with the other drug or with the other body hormones. I imagine that there probably is with leptin, but we can't answer that with the current study. It's a good point.
Okay. That's something that that makes sense. And then was there a reason for why you chose to study this on women? Would the response be similar in men, or how would that differ?
Yeah, so, well, it would obviously be perfect if we studied women and men, but because you'll need such a bigger sample size to look at that, and it was already really tricky to get the sample size that we had, and this study was like, gosh, I guess it was like 300,000 pounds, so it's a lot of money, so that would probably in a way double it. So I think you sometimes have to make pragmatic choices. So we went for women because there's very little research in women so far, and because there was suggesting that there's these sex specific differences. So and then as well, because if you try to do men and women, you will want to maybe control for the sex, kind of the menstrual cycle differences. So we chose women. And as well, because they're more likely to have overweight than men. So it makes them I think, a good population to study. But in a perfect world, we would have looked at women and men and then done analysis based off of sex.